Okay, are you ready? Okay, so first of all, I'm really sorry for the technical difficulty that causing delay. That's completely my fault because I'm in Germany and there are some internet problem here because I'm the guest. I gave the seminar here, but this institute does not allow me to join my own Zoom. So I'm really sorry for, apologize. So I will start. We are ready to go. So, oh my God. Good morning, afternoon, evening. Hola, hello, bonjour, hola, ni hao, konnichiwa, 안녕하세요, and hello all. Welcome and thank you for joining us, the 36th seminar and University of Toronto Day. I am in Jena, on East German city, and I just gave my talk at Max Planck Institute for Chemical Ecology. I thank Professor Sarah O'Connor for inviting me to this exciting institute. She is one of the early pioneer speakers in this seminar series, and I have 8 more weeks of my world seminar tour, and my next stop will be Austria to give my 8th talk of this global non-stop tour. So today, I have the exciting speaker in this Zoom, a little bit delayed, however. So I wanted to introduce our pioneer speaker. It is my tremendous honor to introduce very briefly our pioneer speaker, Professor Mahadevan. He's from Canada. He is a professor from University of Toronto. Because of that, I basically call today University of Toronto Day, and also a Canada Research Chair, one of the most competitive and honor positions. He's the first pioneer speaker from Canada, so he is representing entire Canada synthetic biology today. So please welcome him to the virtual podium. And thank you so much for your time. And even though we have some technical problem earlier today, thank you.
Thank you, Tesuk. I'm just going to start sharing the screen here and see if this works. Okay, so first of all, I have to say thank you to Tesuk. I think what Tesuk is able to do is super inspiring. When I learned of this seminar series, and I was so impressed, and I wish that I had such an opportunity when I was young like Nathan. I think I would have just lapped it up, and I think it's amazing that you're able to put in the time to do this. It's very inspiring. Of course, your globe-trotting talks, again, is very inspiring. I follow your Twitter account. I don't know how many people know this very diligently, Tesuk, and I'm super happy to get updates on your exploits. So I would say keep tweeting. That's all I would say to Tesuk. But on that note, maybe I'll talk a little bit about one area which I'm very passionate about, and this is the role of modeling and optimization and machine learning for synthetic biology, right? And I think this is something that I feel like is a great opportunity to bring in more systematic, more quantitative, more analytical disciplines into synthetic biology. And I think there is a big opportunity. And of course, classically, given that I'm in a chemical engineering department, we think about the main opportunity for synthetic biology in making chemicals or fuels or any kind of therapeutics, that kind of angle. And in all of these processes, right, normally there is a way in which you start with trying to plan a pathway. So for example, if you're starting if you're starting from a particular compound that's known inside the cell and you're going to get a non-natural chemical made, then one of the first things that you need to do is to figure out what is the pathway that can get us there. And these kind of stuff can also be relevant in other disciplines, including, in drug transformations and many other examples, right? And one of the things I want to point out is that there is a significant opportunity for optimization and machine learning in this place, right? So there is a lot of tools in the computational space that's focused on trying to figure out modeling and optimization for predicting these pathways. And these days, especially in the organic chemistry world, there is a significant effort in trying to put together machine learning based rule prediction methods and machine learning based pathway prediction methods, which is then coupled to self-driving robotic labs that predict the chemical conditions and that allow for some sort of a robot iteration to make some of these chemicals in the chemical world. And I think there's a great opportunity for us in synthetic biology to leverage some of these developments, including ways of representing molecules, ways of representing sequences, and copying that over for synthetic biology applications. And the other, of course, clear area is once you have these pathways, right, you need to also invent enzymes. And for this enzyme inventions, there is so much of opportunities in the area of enzyme screening and enzyme engineering. I know that our first pioneer speaker, Professor Francis Arnold, talked about enzyme screening, enzyme engineering, and the whole concept of machine learning based enzyme engineering, which I think is also super important. And these days, people are moving away, I would say, to moving beyond, let's say, sequence-based structure prediction, like in AlphaFold, to sequence-based function prediction, like many others have talked about. And I think there is significant opportunities there for applying protein machine learning for these kind of applications too. But one thing that I'm obviously very interested in and thinking that this is an area that others should also work in is in this area of host optimization, where the goal is to put together more detailed models of the host and use that models for pathway and optimization, strain engineering, etc. Not only that, I think there is also ways in which you can develop models of the synthetic biology circuits, including genetic circuits, and then use those models for optimizing the function of these circuits. And I know that there are a lot of other researchers that have put together thermodynamics-based models for promoter engineering, RBS, like Professor Salis. And these things have been now gone beyond these thermodynamics, physics-based models into data-driven models, and trying to combine them is another option. So basically combining these data-driven models with the physics-based models so that you don't throw out the deterministic components is one area which I think is going to emerge. And I think there are opportunities for young scientists to actually put that and put together a good research program in that space. And lastly, like the classical place, an area where people have started to use or have used machine learning and modeling optimization, etc., is in the bioprocess optimization and control. And I think there is a great opportunity to take some of these omics, genomics based models and combine that with deterministic models of process level models and try to come up with just so-called digital twins or digital biomanufacturing approaches that can help do the scale up and help with data analytics and modeling. So that I think is an area where modeling optimization can help. And in particular, one area where in synthetic biology specifically can benefit from is there are significant challenges in the synthetic biology space that I think can be targeted more effectively using these modeling and optimization tools. And of course, we know all about these things. One of the big areas is the context, right? So if you're trying to put in synthetic biology circuits into a host organism, then the concentration of the host machinery becomes a big problem in terms of translating the synthetic biology tools from different hosts. And again, there are different researchers, Domitila Delvice, who's done some very nice work in the space that I think people should look at. Right? Of course, the other component is if you have multiple parts that you're putting in, then interaction between them is something that you need to look at. Orthogonality, I know many people have talked about this. The other things that is in the metabolic context that's important is also the effect on growth rate. So if you come up with a synthetic biology circuit, you're putting in a probiotic or anything else, right? Then it's really important to understand what happens to the growth rate or the metabolic burden. And not only that, I think there is a lot of people have talked about context, orthogonality, and metabolic burden. There's not as much work in trying to look at the speed of the circuit. So if you want some logic to be executed, then you want to be also thinking about the dynamics of the genetic circuit, right? It's just not the steady state static map, but also how fast things work, right? So for example, if you're considering a switch, then how fast do you go from an on state to an off state? Those things also matter. And lastly, I would say, it's also important for the genetic circuit to perform under robust conditions. So you throw it into a cell, if you change the substrate or if you change the environment, pH, temperature, so then you need to make sure that the cell circuits is functional. Okay? So that's all I would say on the technical side. Then I want to briefly talk about a few things that I learned, which I wish I had known earlier. And these are things, some sort of advice I would say. And one of the things that is the most important, I think, at every level of your career is to ensure that you're able to seek out strong and supportive mentors that are invested in your success and view your success as their success, right? And that's really important, I think. And there are no shortage of examples. There are so many pioneer speakers in this note, including our own organizer, Tesuk, who are examples of people who are able to kind of help you grow, if you will. And the other piece is network so much, if you will. And these days, there are so many other tools that I wish I had, including social media tools that I don't effectively use, but I know Tesuk is a master at using them. And these are things that I would say are an amazing way to learn, how to network, I would say. And the other piece that I feel is very important for me was to try to get some sort of an internship or an industrial international research experience, because putting yourself in an environment that's not in an academic or your own academic environment is really helpful. It challenges yourself in different ways. It challenges your ability to collaborate, and going through that process helps you become a better scientist, I think. And lastly, of course, you want to learn good science communication skills. That is not only talking to your own peers in the scientific community, but also to lay audiences, so that the public understands what you're working on and are also able to maybe even interface with policymakers, so that they understand the value of the work that you're doing. So I don't want to do anything more, and I'll stop here. I'm really excited to hear about Nathan. So Nathan, over to you. I would love to hear more about what you're doing, and I'm really excited to hear what Nathan is doing. I also wanted to thank again Tesuk for this fantastic seminar and just the opportunity to contribute, right? So it's amazing. Thank you, Tesuk. You're an inspiration. That's all I'll say.
Oh, thank you so much. Fantastic talk. And of course, I completely agree with you regarding machine learning and data-driven engineering using AlphaFold, etc. You already mentioned. Also, more importantly, the supportive mentors are the most important for especially junior faculty, young student and postdoc. And also, in fact, I started to use social media starting 2021, not before, but I basically UB, but I became addictive because I kind of found social media could help being connected to all the scientists and researcher worldwide, especially during pandemic.
No doubt, no doubt. I think it's really powerful. I think it's very inspiring for many scientists. One thing I would say on the data piece, though, that I think while machine learning is nice, you shouldn't lose sight of deterministic components, right? I think you cannot throw the physics away, I think. So that's something I want to make a strong pitch. I know a lot of people are talking about machine learning, but I think it's got to be in the context of the physics and
Sure.
determinism.
Absolutely, absolutely. Also, I'm also sorry, my camera installed, a little bit old kind of situation, but I don't want to kind of destroy the other people's computers. I just stick with this current computer. So I'm looking at the other side, but that's why my monitor is the other side. So not, okay, so wonderful. So now the main speaker of today with a longer introduction, Professor Nathan Crook received his bachelor degree in chemical engineering from Caltech in 2009 and his PhD degree in chemical engineering from the University of Texas at Austin in 2014, studying with Professor Hal Alper, one of the pioneers. He pursued postdoctoral study in pathology and immunology at Washington University in St. Louis, School of Medicine from 14 to 17 in the lab of Gautam Dantas, one of the pioneers in microbiome, omics, and antibiotic research. Nathan joined the Department of Chemical and Biomolecular Engineering at NC State University in January 2018, and his lab focused on how to engineer microbial community. He is an absolutely true rising star with a fantastic research portfolio from metabolic engineering to probiotic engineering, and of course the microbiome engineering, basically covering all the hot topic at this moment, I believe. And so I guess, I mean, he might be soon in the kind of tenure, upper tenure. So if you watching as a tenure letter writer, please write one of the strongest letter, because he does deserve strongest letter from anyone in the world. So Nathan, please take it away. And thank you so much for joining us, and both of you today. And then this is now your time to entertain us. Thank you.
Oh my gosh. Thank you. Thank you so much. I hope I can be entertaining and hopefully a little informative. This is an amazing honor for me to be part of this seminar series, just to be in the company of such amazing pioneers. And, the predictive modeling is what is sort of inspiring to me. I'm guilty of testing and doing more experiments than I know how to learn from, and having this ability coming is inspirational. And especially as we go beyond the model organisms like E. coli, we really need this type of predictive stuff. So I'll talk about an organism that is kind of a model organism and kind of not a model organism. I'm going to talk about Saccharomyces boulardii today. It's our favorite microbe in our lab, one of our favorite microbes for a variety of reasons. I hope you'll agree that it's a nice thing to study. And so we're going to talk about it in the context of production of drugs in the mammalian gut. And so before I get started, I want to give a major shout out to my department at NC State. We're a big department, a growing department. We are couched in the RGP area in North Carolina, which is an amazing place for biotechnology, biomanufacturing. Our students have no problems getting jobs in the area. So it's a great place to be. We're also situated between the beautiful mountains, the Appalachian Mountains, and the Atlantic coast. So it's a nice place to live for a while. So if you're considering a place to go, this is a great place to do it. And I have amazingly supportive mentors here. So it's a nice collegial environment as well. My lab is exceptional. I have amazing graduate students. Everybody from Ibrahim to John is a graduate student. Katie through Sean are all undergraduates. And none of this work, it would have been possible without them. You can only do so much as a PI. I cheerlead, they do the real work. And I think you'll see the quality of these individuals from the data that I'm showing. So our inspiration for this work, we're all chemical engineers, and we think about ins and outs in a system. And so the gut microbiome is a really great system with lots of ins and outs. For example, the dietary material flows in, and this is undigested fibers, proteins, bile acids from the small intestine. And its microbial community converts it into an amazing variety of value-added products, right? Energy molecules, immune antigens, even some vitamins are produced by the gut. This gut microbiome does chemistries that our own body simply can't do. So, just by the factor of how many genes are in our genome, we have about tens of thousands of genes. But the collection of microbes in the gut has many more genes. They can do chemistries in their own cells that are incompatible with the biology of our own cells. And so you can think of them like this sort of tunable bioreactor that we have access to in our own large intestine. The great part about this bioreactor, though, is that it's not fixed, right? So this bioreactor can change naturally depending on the food you eat, how old you are, your exposure to molecules in your environment. But it can also be changed through directed means. For example, you can take a probiotic and add a new genome into your personal bioreactor. And so we got really excited about this idea because, what if we could look and try to optimize the chemistry for particular applications? And so in the beginning of this talk, we'll talk about one particular microbe in this community, this probiotic yeast, and its ability to produce a vitamin that might be accessible to the host. So Saccharomyces boulardii, what is it? Well, it's a eukaryote, and as a microbiome person, eukaryotes are one of the sort of understudied components of the gut. One of the reasons why is because the gene fraction, right? So if you extract the total genomic DNA from the gut microbial community and just assign it a taxonomy, you'll see about 0.1% of it is eukaryotic in origin, non-host eukaryotic. And so that means, okay, maybe this is not a major component or a major player in the gut. But one of the things that is interesting about eukaryotes is that their cell size is much larger than a bacterium. So cell size divided by DNA would indicate that the mass fraction of eukaryotes in the gut is not insignificant, in the tens of percents perhaps. And this is sort of supported by fish images of eukaryotes lining the mucus layer of the large intestine. They're there, and they do chemistries that we need to understand. Boulardii in particular is a fungal probiotic. It's a relative of Saccharomyces cerevisiae, and it has a sugar-coated cell wall that it loves to bind to pathogens with, and it's available and generally regarded as safe in your local pharmacy. So it has a nice track record of safety, and it's a nice platform by which to build additional gene functions into. The nice things about Saccharomyces boulardii that we are really excited about is that it is genomically similar to Cerevisiae, so all of our knowledge of Cerevisiae might be applicable to this strain. It also grows at body temperature. So 37C is its optimal growth temperature, which indicates that it's a professional gut colonizer. Our goal in this work in our lab is to engineer boulardii to deliver biomolecules to large intestine. We think it's nice from a biomolecule perspective because yeast is a workhorse of protein production. Cerevisiae is a workhorse of protein production industrially. So maybe we can port some of that ability into the large intestine too. And so what we wanted to do is produce this molecule beta-carotene as our first test case. So this is our work that we just have described in ACS Synthetic Biology recently. So I'll briefly go over what we did. So beta-carotene looks like that. It's got a lot of double bonds that make it orange. It's a precursor to vitamin A, more importantly. About 12 equivalents of beta-carotene make one equivalent of retinol, which is your vitamin A. We're interested in this molecule because vitamin A deficiency is a big problem. A lot of people in the world can't get enough of this molecule. And so perhaps if we could engineer their personal bioreactors to produce this molecule, we could alleviate some of this problem. Another benefit of a lot of carotenoids is that they are anti-inflammatory. In particular, acetyltransretinoic acid, this is a derivative of beta-carotene that's cleaved down the middle. It attenuates colorectal cancer when it's delivered to the large intestine. So this is just a picture from this study that delivered this all-transretinoic acid into a mouse gut that was prone to develop colonic tumors. And so vehicle has more tumors than the all-transretinoic acid group. So it's beneficial in more ways than just the vitamin A nutritional component for it. So it's an interesting thing to do for the gut by itself. The great thing about vitamin A or beta-carotene is that it's colored. And so we can use the ability of yeast, right? Yeast has this amazing ability to combinatorially optimize pathways. And what you can do is get a whole plate full of all different organizations of this beta-carotene pathway. If you zoom into here, you see each colony on this plate has a different shade of orange. And so my students, this is one of the first things they did, is they got this library of beta-carotene production to be made. And they scoured this plate, picked individual colonies, and they got a library of diverse beta-carotene titers. We were able to figure out what the pathway organization was for all of these, and we were able to figure out the beta-carotene production rate of all of these strains. We chose one of these isolates, isolate number 23, as our isolate of choice to deliver into mice to see if it would colonize. So these were germ-free mice, mice that were grown in completely sterile conditions. They're grown in these plastic bubbles that have no ability to exchange microbes with their surroundings. They're born germ-free, and they will remain germ-free until you give them the microbe that you're interested in studying. And so if our yeast cannot produce vitamin A in this context, then it's hopeless. So what we did is we started with this model. We had two groups of germ-free mice. One, shown in black here, was mice that were colonized with wild-type boulardii that does not produce beta-carotene. And group B were mice that did produce beta-carotene. We wanted to know if there would be any difference in their fecal beta-carotene titers. The first thing we wanted to test, though, was, does this additional beta-carotene produce a colonization defect or a burden? This is what we talked about at the beginning of this seminar, and it's really, really important. And so in black are the wild type yeast, and in sort of peach colored are the engineered yeast. And we had a little bit of a growth defect. It wasn't as substantial as we were fearing, but it does stably colonize this mouse model, which was great to see. On the other y-axis, on the left-hand side, you can see the milligrams of beta-carotene per gram of feces. And between the two groups, there was, in fact, a substantial difference in the amount of beta-carotene that these yeast were able to produce. It grows up until day 4 and then reaches sort of a steady state. And it's above the natural levels of beta-carotene that exist in this mouse's diet. So we're actually producing about half, if you go through the math, about half of the recommended daily allowance of beta-carotene, of vitamin A in these mice using this sort of first experiment. So we were really excited by this. It indicated that this yeast is able to produce a value-added product continuously in the gut of the mouse from undigested material. And we started thinking about microbiome engineering in general in a more sort of chemical engineering-y way. So if you wanted to draw a sort of pathway map of what you might want your gut microbiome to do, it might look something like this, where dietary fiber comes in and different microbes based upon the health of the host turn on the production of different metabolic modules. Maybe there are strains that tune the production of energy you can get from your food, or produce drugs that you might need based on health host markers. So in order to realize this dream, we wanted to conceptualize this into a couple of parameters that reduce the complexity of the gut microbiome, right? So if you want a microbe to produce something in the gut, you need a few things. One is a drug that's potent enough to have an effect on the host. Another is a chassis that can produce the drug at the therapeutic levels. Finally, you want a chassis that can stick in the gut for as long as you need it to. Maybe you want a microbe, for example, in green, that doesn't stick around very long because the condition you're treating is an acute condition. On the other hand, maybe you have like for example, a cancer that's going to stick around for a while, and you want a bug that is stable and able to colonize next to that tumor and keep it in check for a long period of time. And so we're starting to think about key parameters to sort of go after one by one in our engineered probiotics. And so I'll talk about each one of these in series. The first of which is drug potency. And the drug I'm going to talk about is targeted toward this particular pathogen, C. difficile. C. difficile, you've probably heard of before. It makes these really bad gut infections. It's classified as urgent by the CDC. It's got a spore stage that's really tough to eradicate. Spores, when it gets into a gut that's been treated with antibiotics, those tend to germinate because there's lots of nutrients available. And once it germinates into a vegetative cell form, it secretes a toxin that destroys your gut lining and can lead to movement of material from the gut into your body and movement of material from your body into your gut. It makes for a very bad infection. Not only that, but if you kill the vegetative cells, it's hard to kill the spores. The spores are intrinsically resistant to antibiotics. And so you have a 20% relapse rate of this disease, even if you can treat it once. So there's a definite need for new treatments here. So we wanted to target the virulence factors of this C. diff. So it's two of the major toxins of C. diff are toxins A and toxins B. They're structurally similar proteins, but they have different activities and slightly different specificities for their target. There are large proteins. They have multi-domain proteins. You're shown here in these different colors. All of those components from C, D, and B are involved in getting the warhead domain A into the cell. So it has to bind to the cell surface, get into a vesicle, translocate itself out of the vesicle, and then into the cytoplasm where its molecular target is. Now, its molecular target are these Row and Rap GTPases. It adds a sugar molecule, glucose residue, onto a particular location of these GTPases that inactivates them. Once these GTPases are inactivated, the cytoskeleton breaks down, and you have breakdown of the tight junction between your cells on the wall of your gut. So this is how this toxin works. And if you could inhibit the toxin, maybe you could synergize with antibiotics or inhibit the infection process on its own. So here's the team we developed at NC State to try and access this really complicated and fascinating infection. It's composed of myself and my student Dennis, but also Stefano Menegatti and his two students, Carly and Ethan, involved in peptide discovery, Carol Hall that can do computational peptide simulations, and Scott Magnus at UNC, who has this really nice cell-based system that I'll talk about to assay the ability of these peptides to work. And so as a collaborative team, we're trying to discover peptides that selectively inhibit the active site of these toxins and deliver them using our probiotic yeast. So we came up with this really clever screening strategy, and this is not my idea. This is a collaboration between Steph and Carly. And what they thought of was this idea of coding beads with peptide candidates. So what they can do is make libraries of tens of thousands, hundreds of thousands of beads, each of which is coded with the same peptide. So bead A has peptide Y, bead B has peptide Z on the surface. Now, what we can also do is color the toxin in red. So if our beads bind to the toxin, then the bead should turn red. But that's not good enough for us because there's a lot of places that a peptide could bind on the surface of this toxin. It could bind to the active site, which is what we want, but it could also bind another place on this toxin, maybe the crop domain or a domain that's not involved in the actual transfer of the sugar residue onto the target protein. So they came up with a clever secondary screen. One is, and this is to label the substrate of this toxin. UDP glucose is a substrate, and they added a fluorescein molecule to the end, with the notion being that if the peptide binds to not the active site, then the toxin can still bind to its substrate. And so what you'll get is a bead that's both red and green. And so it's sort of like a Christmas show over here. What we want to do is sort the beads that have a high red binding and a very low green binding. So this is a really cool idea, and it required the production of a microfluidic device to, in an automated fashion, take these beads and sort them, one way or the other based upon their fluorescence profiles. And Ethan, in co-advised between me and Steph, was able to get this device to work. So we were really excited. We screened thousands of beads through this method. And I'll show you some examples of what we saw. So what you see in this picture, sort of on the red channel, is the toxin binding. And you see that a lot of these beads have sort of a halo effect of the red on the outside of that bead. And that made sense to us because this toxin, the size of the toxin, the hydrodynamic radius, is much larger than the pore size of the beads. However, UDP glucose, the fluorescein labeled version, is much smaller than the pore size of these beads. And so we had to optimize how long we were incubating the UDP glucose so that we just didn't get diffusion of that UDP glucose into the bead and cause everything to turn green. So after getting that taken care of, we were able to find beads that had a high red fluorescence and a low green, which shows the most promising out of these. And we sequenced them using Edman degradation. Now, in a collaboration with a computational group, we were able to get the peptides that inform the design of peptides computationally as well. So I'll show you both the results of these experimentally designed peptides and some computational designed peptides. And I'll show you the results in the context of a cell-based assay that the Magnus Lab developed. So what you're seeing here is a graph that has no data on it yet, but it measures resistance across a monolayer of cells. Now, these cells are primary human colonic cells. These are cells derived from a human donor. The donor has passed away, but they donated their organs to science. Scott Magnus can take these cells and grow them in monolayers for a long period of time, add toxin to them, and see if they get intoxicated by this protein. That should reduce the resistance across the monolayer. And so if a cell is healthy, you should see a high level of resistance. If it's not healthy, you should see a low level of resistance. And so I'll show you the data on this panel and walk you through it. So when you have healthy cells in red without anything added to them, the resistance gradually declines over the period, over about a week in which this is performed. If you add toxin 2 hours later, you see a drastic decline in the resistance as you would expect. Those tight junctions are breaking down. However, two of our peptides, S1, which is a computationally derived peptide, and LS5, which is one of these library screened peptides, both mitigated the toxicity of TCDA. Some of them, this computationally derived peptide, informed by our screening results, performed had cells that were even more resistant than our cells that were untreated with toxins. We're really excited to go forward on these peptides and study them further. So I've talked to you about how we're looking at drug potency in this work. I'm going to next talk about how we're engineering the productivity of our strain for higher production of this peptide. So in order to make this better, Dennis, my student, took a deep dive into the protein secretion mechanism of yeast. And so this is shown here in this cartoon. There's multiple steps in this process, getting from the nucleus into the endoplasmic reticulum, the various quality control steps in that space, getting it to the Golgi, and eventually out to the exterior. And we were able to identify the boulardii homologs of all of these well-studied yeast enzymes. We were able to make knockouts in each one of them sort of individually. So she was able to go to our collaborators group at Denmark Technical University, DTU, Jose Luis Martinez Ruiz, who has a really nice platform for doing high throughput bioreactor studies. So we could take our wild type yeast and study how much peptide it makes. And in this case, our wild type yeast was making 500 nanograms per microliter of our peptide, 500 mg per liter, which is already really good. If you try to do this experiment with bacteria, they have about an order of magnitude lower expression of peptide-sized things. However, when you looked across all of these knockouts, what we saw is a wide diversity of increased, sometimes decreased peptide production. And in fact, several of these, in fact, the four shown on the right, really improved the production of our peptide, which we were very pleasantly surprised by. Unfortunately, I can't tell you the actual names of the knockouts because of IP reasons, but if you're interested, please do let me know. I'll be happy to share them with you offline. We were really interested in these last four because they all seem to really improve the secretion of our peptide without impacting the growth very much. So what we did is we made combinations. So here I'm just showing you an abridged version of all of these four knockouts, and I'll show you the combination of 3 and 4, and then we took a real Hail Mary and made all 4 together. And so the 3 plus 4 had a modest increase in peptide production, which we were very excited about. However, when I heard that they had made all 4, I was like, wow, you really went for it. I don't think that'll work, but it actually did even better than the 3 plus 4 together. So this strain can make about 5 grams per liter of a peptide that we have designed computationally. And that is well within the therapeutic range of this toxin inhibitory peptide, which we're really excited about. So what we're trying to do now is see whether this strain works the same in gut-like conditions and in an actual mouse. Speaking of working in an actual mouse, what we want to make sure is that the promoter activities of these strains are actually sufficient to work in the gut, right? It would not be beneficial at all if our strong promoters that work well in culture don't turn on in the gut context. So to see what's going on here, we took our yeast and we put it under different gut-like conditions, plus or minus oxygen, stationary phase, or exponential phase, and we did RNA sequencing. And just to show an overview of the data that we can get here, what you're seeing is the RNA sequencing or a heat map of the transcript levels. We have the different cultures we chose on the top, and the different genes that were significantly changed in their expression on the side. And for example, these sets of genes shown in this box have a highly upregulated expression in only anaerobic settings, both in exponential and stationary phase, but only anaerobic is when they turn on. Now, I'm not showing the gene names here for the sake of space, but these are actually largely uncharacterized genes, even in Cerevisiae, which is really exciting. It indicates that there's new biology going on in these yeasts under anaerobic conditions that we can harness for perhaps gut-specific production of our therapeutic. So that's our brief study on the productivity of our chassis. And now I'll say briefly about how we're thinking about how to engineer the abundance of our chassis in the gut. This is a hard problem for a variety of reasons, not to mention the fact that the gut is a very complex environment. Probiotics have an interesting life cycle. They start out in a bioreactor or in a food product, and then they're sort of dumped into an environment that's very, very different, right? This is a gut that has multiple selective pressure. You have immune systems, you have physical flow, you have metabolism of the endogenous microbiota that could be inhibiting you. If you're a bacterium, you have phage. And so which of these features most limit your survival is not really known. So perhaps what's going on is that these probiotics are actually evolving. They change their genomes when they live in the gut. They can reproduce. And what we wanted to know is which of these functions are under selective pressure. Is it functions involved in nutrient metabolism, or is it adherence, for example? We can also ask questions about the diet or the microbiome state of the host. So we've already done this experiment as a postdoc for the context of probiotic E. coli, and we're trying to do something similar in the context of probiotic yeast. Now we're starting to do this based on an experiment we did with wild type yeast that we did for our ACS synthetic biology paper. We took this yeast and we put it under 3 different mouse conditions. In the gray, what you're seeing is a germ-free mouse, and you're seeing this colonization level over time. And you can see that this yeast very stably colonizes a germ-free mouse. However, if you take a conventional mouse and you pre-treat it with antibiotics, you get a much lower level of colonization. Actually, if you don't treat them with antibiotics at all, you would see something like a very vertical line here. It doesn't colonize a normal mouse very well at all. You need antibiotics to enable it to gain a better foothold. So what we're trying to do is take isolates from this experiment and sequence them to identify mutations that are occurring. And so our first foray into this is this 30-day time point from a germ-free mouse. That's plenty of time for yeast to evolve. And we're doing whole genome sequencing of this yeast strain. Now, what's very different from a probiotic or a bacterial probiotic is that this yeast is diploid. It has 2 copies of each genome. And so what we saw were a lot of mutations that were loss of heterozygosity, where the initial wild type strain had 50% abundance of a particular change. And when you sequence the evolved isolates, you got 100% concordance of that mutation, indicating a recombination event had occurred. This is a well-known mechanism of evolution in eukaryotes. We also saw things like a small gain of heterozygosity, where in a particular location, a single SNP changed in one of the arms of the chromosome. And that also sort of made sense to us as a mechanism because you don't need very many things to happen in order for that to occur. What we're very confused by in this data, and we've only analyzed a bit of it so far, so that's why I don't feel confident telling you all the mutations we saw, is that we saw several isolates, in fact, all of our isolates had large gains in heterozygosity in the same location of their genome. So these were hundreds of kilobases long, and they all had, and they had collectively about 50 to 100 single point mutations across 100 kilobases. So it indicated something really unexpected was happening here. We're not really sure how all those mutations could have happened in those strains. So we're going to be sequencing earlier time points to identify if this is sort of a gradual process or an all-at-once process. We've considered things like mating of this yeast, but there's no other yeast in the cell in this model. So that doesn't make as much sense to us either. So stay tuned for the answer to this. We're sequencing more isolates, and hopefully we'll figure out the answer to it soon. So that's collectively our current experiments into how we can maybe make this probiotic yeast into a workhorse for the large intestine. We've explored drug potency, the productivity of this chassis and also its abundance. But this is a general paradigm for any probiotic that you, or any gut commensal that you might want to engineer. Human gut, roots, etc. These are the parameters you want to look at. So I want to thank you very much for listening to this talk. I hope you learned something. I hope you were entertained. I have the main students who worked on the work I showed you here. We also have other students in my group who work on fascinating projects in other areas, mostly on the bacterial engineering side. And I want to thank the funders for this work, especially because they take a risk on somebody who is young and who has crazy ideas like me. Keep doing that. If any of you are listening, keep funding high risk work. And yeah, I'll thank you and take any questions you have. Oops. Oh, yes.
Yeah, great talk, Nathan. I really like the whole approach to trying to keep SB as an option for engineering probiotics. One question I wanted to ask was, like, you said that, you know, when you actually combine the 4 mutations, which we don't know what they are, but it's okay.
Yeah.
You saw a synergistic effect, right? And
Yeah.
I'm surprised. I've never seen something which is so linear and synergistic, right? So I'm trying to understand, like, maybe you can mechanistically explain why you think that might have happened. If you think any ideas, you know, just fascinating though.
Right. And so, you know, of course I can tell you more about it offline, but we think what's going on with these knockouts is that each knockout hits a particular, you know, they each reduce secretion a little bit, right? And they hit independent modes of secretion, right? And so because they're independent, the combining them can make sense, you know? And so in this case, what I was actually very surprised by was that when they did 3, right? So they did 2 and 4 here. They also did 3, and that actually killed secretion, which is very unexpected. I thought, okay, linear increase, you know, this is gonna, you know, we'd see a nice trend there, and choosing a subset of 3 killed it. And so we're yeah, it's amazing. This thing is reproducible. We've tested it this way and that to make sure we're seeing the right thing, but it was very unexpected for us too.
Really cool. So can I ask you one more question, if that's okay? I think maybe since there's nobody else, I might take a liberty to ask. Right. So one of the things I wanted to understand is, in these probiotics, right, how effective are they in keeping, like, you know, in colonizing the gut, even when the diet is changed, right? So you stop introducing this SB and diet is changed. How long do they hang around in the diet, right? You think that there is a kind of PFR type action that will flush them away, no?
Yeah, and so that's exactly what happens, right? In this strain, in lactobacillus and probiotic E. coli too, they don't last very long in the gut. They're not professional colonizers. There's a really impressive study done several years ago where they took commercially available probiotic consortium, I think it was VSL3, and they put it into people consuming different diets. And it was amazing. You know, the diet of the individual, the genotype of the individual, all played a role in the residence time of these strains. So if we think about producing this as a therapeutic, that's a kind of a problem, right? I mean, the way around that is to say, well, we're going to make short-lived therapeutics until we figure this out, and that's fine.
Yeah, but it's maybe a feature, right? Because it's not going to harm the body or something like that, because it's getting out, no? So it's not terrible, I think. But I understand what you're saying, you know? Yeah.
I you know, the pie in the sky in me wishes I could figure out what causes a strain to be a permanent colonizer. But there is this like issue where maybe we don't want it to be there, exactly like you said. So it would be nice to at least tune it, right? It'd be nice to have a knob you can turn that would allow you to stick around longer if you needed it to.
Maybe interact with the mucins or something like that, right?
Yeah. I think the mucins are going to play a big role.
So Can you hear me?
Yes.
Oh, I have the mic and camera issues. I couldn't join you, I mean, earlier. So, but fantastic talk. I mean, I really enjoy. And then also thank you. I mean, you know, basically, you know, you asking why I'm away. So that's kind of fantastic. Though I have no idea what's going on in this, you know, situation. But probably right now I'm the East German city, so old, you know, communist kind of, you know, remnant, kind of still kind of not that happy with me because I'm from Korea. Korea still have the two separate country, unfortunately. So thank you so much, you know, Krishna. I mean, you know, you kind of make this show continue while I kind of away, and I couldn't join, you know, you, I mean, by camera. So I actually have one question. So you beautifully describe the probiotic, you know, engineering work, and as you know, I'm working on E. coli inhibitor, and then some other people also working on, you know, lactate. Basically lactate producing, you know, bacteria. So many different probiotic. And as you know, probiotic, you know, not that much able to colonize. And you know, another something crazy thing I kind of thinking about is you may want to have mixture of probiotic, including your, you know, host, E. coli, and, you know, lactic acid, you know, producing bacteria, and then somehow in a consortium that might have some potential synergy, but we are not sure, of course. And can you kind of share your perspective or insight into those things? Because I kind of thinking about even some project, big project, you know, all the probiotic engineer or scientists coming together, kind of asking huge money from some organization or government. That might be something fun, right? So I just want to hear from you.
Yeah, so I think that each, you know, there's sort of a reason why our microbiomes are so diverse, right? Every microbe that is there is good at something, you know, and so I think that in terms of our engineering, we should take the same approach. You know, this yeast might be great at producing proteins, right? But maybe it doesn't colonize where in particular locations where this lactic acid bacteria can colonize, where E. coli can colonize. I know that E. coli, you know, it can produce way more protein intracellularly than yeast can secrete extracellularly, right? So if your thing that you want to produce is an intracellular protein, E. coli could be the way to go. And so I think there needs to be a study on sort of the like, what are the, you know, comparing head to head on the same types of therapeutics, what is, you know, the best strain to use? And it can go beyond these probiotics too. I know people are doing fascinating work in the Bacteroides space, bifidobacterium, right? There's lots of cool things to do there. If I need to spitball even more, right, we need engineering tools for these strains. They're hard to transform, right? These strains, it's much easier to make an E. coli strain than it is a bifido strain for whatever reason, right? We need to understand what the limitation, what fundamentally limits our ability to engineer these. And again, we also need a journal. I think we need a journal for engineering probiotics. I think that'd be amazing.
Sure. Of course. I mean, you know, I completely agree with you. That's fantastic insight. We also need to discuss more. I mean, that's an exciting field. And then actually the NIH and other agencies very interested in this kind of field. You know, that's kind of good thing for us. So I guess, I mean, we a little bit kind of over because of my technical problem earlier. So I apologize again for everybody actually who tried to join earlier, some audience. I kind of got some email from some of that, and then unfortunately they couldn't join. But still, you know, some of you still stick around today. So thank you so much. Also, I'm apologize one more time. This is part of the problem in the world tour because I end up kind of have some delay of train, and that kind of make me wait two hour or 2 hour delay, and then miss my connection. But show must go on. I mean, regardless of those kind of problems. So also thank you so much, both of you, joining again. Even though you have the technical problem not coming into the Zoom meeting, so finally everything worked out. And that's all about life. I mean, research, all the trouble.
Yeah.
And at the end of the day, we, somehow succeed. So and thank you so much. I mean, and then sharing your insight, especially, Nathan, beautiful talk. And also, I couldn't say something before, but also Krishna, I mean, basically you give very, very nice overview of entire synthetic biology sort of process from the upstream to downstream. So that's fantastic. So thank you so much. So I wanted to close because we already kind of passed 10-minute normal regular time. So thank all for joining and staying today. We will meet again next week on June 9. Hopefully there's no problem. Thursday, the same time, the same Zoom link, but I will be joining from Belgium after my 9th talk at Leuven. We will have Professor Domitilla Del Vecchino at MIT and Professor Jeff Cameron from University of Colorado Boulder. Excuse me. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with today's speakers. I will promote you to panelists who can speak and show your handsome and pretty face. And thank you so much. I'm stop recording. Let me stop recording. And that way we can freely talk.